Hello! The learning outcomes for this presentation are to 1. Describe the importance of functional groups in organic compounds. 2. Briefly state what lipids are and give some examples of different types of lipids. And 3. Describe the structures and functions of lipids. Lipids are organic. Organic molecules have a carbon-based skeleton. This means that the core of the molecular structure is primarily composed of carbon. Organic molecules also contain hydrogen atoms and usually contain oxygen as well. These types of molecules are covalently bonded and have functional groups that determine what type of lipid it is and how it reacts with other molecules. The macromolecules of living organisms, including carbohydrates, proteins, lipids, and nucleic acids, are considered organic molecules. Lipids are mainly hydrophobic molecules that include oils, waxes, fats or fatty acids, eicosanoids, glycerides, steroids, phospholipids, and glycolipids. We will go over each of these types of lipids in this presentation. Fatty acids are long chains of carbon and hydrogen with a carboxyl group, COOH, at one end. They are relatively nonpolar, except for the carboxyl group. A fatty acid is either saturated, that has single covalent bonds only, or unsaturated if it has one or more double covalent bonds. The presence of a double bond causes a sharp bend in the molecule. A structural formula and computer models show that a saturated fat is a straight chain of carbon atoms joined only by single bonds. In an unsaturated fat, two of the carbons in the carbon chain are joined by a double bond. There is a kink or bend in the molecule at the site of the double bond. It is important to remember that fats and fatty acids are types of lipids. Another type of lipid are the eicosanoids. These molecules cannot be synthesized by the body, so they must be obtained from the diet. They are derived from a fatty acid called arachidonic acid. Examples of eicosans include leukotrienes, which are molecules that play an active role in the immune system, and prostaglandins, which contain short-chain fatty acids and act as local hormones to regulate functions such as inflammation. Here is a structural diagram of prostaglandin. It contains 20 carbon atoms and a 5-carbon ring. Glycerides are another type of lipid. They contain fatty acids attached to a glycerol molecule. A monoglyceride has a glycerol molecule plus one fatty acid. A diglyceride has a glycerol molecule plus 2 fatty acids. Lastly, a triglyceride has a glycerol molecule plus 3 fatty acids. Triglycerides are also called triacylglycerols or neutral fats. They probably sound familiar because much of our adipose tissue is composed of triglycerides. Because they make up much of our fatty tissue, they have 3 important functions. 1. They serve as an energy source. 2. They insulate our bodies and regulate body temperature. And 3. They protect our vital organs from injury. The formation of a triglyceride involves the attachment of fatty acids to a glycerol molecule through dehydration synthesis. In this example, a triglyceride is formed by the attachment of 1 unsaturated and 2 saturated fatty acids to a glycerol molecule. Steroids are another type of lipid. They contain a 4-ringed carbon structure and have a variety of functional groups which affect how they function and the type of steroid they are. Examples of steroids include cholesterol, which is a component of the plasma membrane of all of our cells, sex hormones such as estrogen and testosterone, steroid hormones such as corticosteroids and calcitrol, and steroid derivatives such as bile salts. You can see from the examples that the functions of the steroids varies widely, but each play important roles in maintaining homeostasis. Looking at the structure of these steroids, you can see the 4-ringed carbon structures and how each individual steroid differs in the side chains attached to the carbon rings. Phospholipids and glycolipids are another type of lipid. Both of these lipids can be synthesized by our cells, so we do not need to acquire the molecules through diet. They contain a diglyceride attached to either a phosphate group for phospholipids or a sugar for glycolipids. Generally, both have a hydrophilic head and a hydrophobic tail. They function as structural lipids as they make up the structural composition of the plasma membranes. In this slide, we can see a model on the left and the structural formula on the right of lecithin, a type of phospholipid. There is a non-lipid group in the head of the molecule, followed by a phosphate group, then a glycerol attached to two fatty acid tails. One fatty acid is saturated and straight with no double bonds, and the other is unsaturated with one double bond and appears bent. In a glycolipid, a carbohydrate is attached to a diglyceride. The carbohydrate here consists of two 6-carbon rings attached to the glycerol head, which is attached to two fatty acid tails. One is saturated and straight, and one is unsaturated and bent. When large numbers of phospholipids and glycolipids are in water, they form micelles, with the hydrophilic heads facing the water molecules and the hydrophobic tails on the inside of each droplet. In water, spherical structures form with the hydrophobic tails tucked inside, away from water, and the hydrophilic heads on the outside, contacting water. In this presentation, we learned that lipids are organic molecules that are mostly made up of carbon and hydrogen atoms that are covalently bonded together. We also learned that there are many types of lipids and that the functional groups determine the molecule and its reactivity. Lastly, we learned how lipids are mainly hydrophobic molecules and how this characteristic trait of lipids affects the function and reactivity in aqueous solutions inside the body.